Anyway. All right, well, thank you so much for allowing me to present to a different group the work that I'm doing on red cell rheology as a tool to assess novel therapies in sickle cell disease. And since this isn't totally sickle cell audience, I feel obliged to put in this pathophysiology slide, although I feel like everyone at Emory probably is very familiar with sickle cell disease. That's one of the reasons I love Emory. But of course, it's the first disease for which we knew the abnormal protein, the abnormal nucleotide change. And what happens is subjects put a valine in place of a glutamic acid into their hemoglobin protein. And this results in a hydrophobic residue in place of a hydrophilic one. And to bury that hydrophobic residue, we create these polymers or stacks within the red cell. And this makes the red cell rigid. It makes it very poorly deformable through the microvasculature. And there are other effects too. There's chronic inflammation, hemolysis, stickiness of not only the red cell, but the white cells as well. But for a long time in sickle cell, we were very focused on the red cell, and that's something that I'm guilty of as well at times, but trying to think also about the interaction of the red cell with the endothelium. And this one little change, which helped protect carriers from malaria, produces a myriad number of clinical complications, including stroke, pain crises, bone damage. Almost every organ is affected. And so it's been exciting to be in this new era of therapies for sickle cell disease, because for a very long time we only had hydroxyurea, often abbreviated HU, and it causes the individual to produce more fetal hemoglobin, a hemoglobin with a beta globin from a different gene not affected by the sickle mutation that dilutes out and prevents polymerization of the sickle hemoglobin. That's what was our tool for almost 30 years. But now we have crizanlizumab, and monoclonal antibody that blocks the adhesion to the endothelium. And we have voxelotor, which binds to the sickle hemoglobin, keeps it in its oxygenated state, which helps prevent that polymerization. And we have L-glutamine, which may have some antioxidant properties. And of course, we're entering a new era of gene-based therapies. One may be FDA approved very shortly. And so with all of these different therapies that aren't so straightforward as hydroxyurea, with hydroxyurea, you knew if you got fetal hemoglobin, you were making a positive impact. But now we need other tools in order to assess these therapies that may not have any drug change at all, or any laboratory value change at all, like Endari, for example. And we need a tool with which to compare all of these different gene therapies and to determine whether we've achieved a functional cure. So rheology is the study of the flow of matter, and the rheology of sickle cell blood is very abnormal. The blood is very viscous for its level of hemoglobin. So an individual with a hematocrit of 21% with sickle cell has the same viscosity or overall whole blood thickness as a normal individual, a hemoglobin AA individual with a hematocrit of 45. So our patients really have to be anemic, or else their blood would be like motor oil, it'd be like sludge. So why is their blood so thick? Well, their red cells are denser. They're packed with this abnormal, poorly deformable hemoglobin. And they're also more adherent and have other abnormal properties that are a little more difficult to study outside of the tools I'm going to describe. So we have several devices to measure red cell rheology. And I'm kind of shifting in my mind to thinking about it more like red cell physiology. So one of them I've not used, but I'm excited to use because it lives right near my lab in the ECC. But what I've been using at Baylor is a cone and plate viscometer, quite an old tool. You put the blood in this cup, and this is the cone. Looks like a flat plate, but it actually has a little angle. And it kind of shears or smears the blood in the cup, and that's the way you tell how thick it is. And you can set it to different shear rates to mimic different aspects of the circulation, venous versus arterial. But it's, of course, very removed from physiology. You're just looking at the whole blood out of context in a way. I have an ADVIA hematology analyzer, which gives me a CBC and a retic count and all those good things, but it also calculates the % dense red blood cells. There's an older way to do this using a phthalate gradient, but the this device does it automatically and with really good reproducibility. I have an ectocytometer with oxygen gradient, which lets me see the changes in red cell deformability or stretchiness over a range of oxygen concentrations. And this is an important improvement from the existing ectocytometers that just looked at the oxygenated red cell, because deoxygenating the red cell is key to unearthing its abnormalities. And so you get a lot more information once you can deoxygenate and reoxygenate the red cell. I've been beginning to work with microfluidic devices. I had a collaboration in Houston with an engineer who produced them. And now I'm working with Wilbur Lamb's lab on using his device. And I'm getting more and more familiar with it. The HEMOX, again, I haven't gotten to use it yet, but I anticipate adding it to this arsenal. Measures the oxygen affinity, which I'm sure is quite useful for agents like voxelotor. So the ectocytometer, again, it measures the deformability of the red cell. The red cells are injected into this viscous solution, so they're kind of floating there, but it is making a population level measurement, not a single cell, even though it can kind of look like you're looking at a single cell. And a normal red cell, when you apply stress to it, either through salt concentrations or through shearing, stretches nicely, gets its nice oval shape. A sickle red cell, though, does not. It's chock full of the hemoglobin polymers, and so it cannot deform and stretch the way a normal cell does. It still has these angles or corners. And it's less so when it's oxygenated. It's even worse when it's deoxygenated, again, because that deoxygenation potentiates the polymerization of the sickle hemoglobin. So here is a little video, particularly watch the blue box. So what's just happened is we're measuring the deformability of the red cell as we drop the oxygen concentration. And there comes a point where the deformability drops precipitously. That's termed the point of sickling. And then you can see that the red cell's deformability declines. Lower numbers are worse. Lower numbers indicate more rigidity. And then we reoxygenate the red cell. And you can see that the flexibility improves and it returns back to about its prior state. So this gives us 3 biomarkers. You could claim more than that, but what I mostly look at is the elongation index maximum, the stretchiness of the red cell when it's fully oxygenated, when it's at its best, the point of sickling, that oxygen concentration at which the hemoglobin begins to polymerize and the deformability begins to worsen, the elongation index minimum, which is the red cell at its very worst. And some people also look at the difference between the elongation index maximum and minimum, and there may be some utility to that. I'll show you the data on that later. So you could give me a tube of blood and not tell me where it came from, and as long as it's one of these genotypes, and I should be able to tell based on the curve it produces on the ectocytometer what genotype that patient had. So a hemoglobin AA individual gives you a straight line. There's nothing to sickle. A trait individual, hemoglobin AS, can tolerate a lot of deoxygenation before it finally sickles a little bit. And you can see there's very little difference between the elongation index maximum and minimum. It just gives you that little loop, because there isn't that much within that red cell to sickle. But a sickle cell individual has really poorly deformable red cells, even when fully oxygenated, even at its best state. Its point of sickling, depending on the patient's disease severity, treatment, therapy, will be somewhere in this range. And at some point, some personal PO2, the red cell will begin to sickle, and you'll have worse and worse deformability until you hit your elongation index minimum. And so we have, of course, these long used accepted therapies that we can see their improvement using this tool. You can take a patient and titrate up your hydroxyurea to maximum tolerated dose, and at every visit, look and see how much their blood has improved, their deformability has improved. So starting, at that typical hemoglobin SS curve, you can see that as you increase the hydroxyurea and you start to make fetal hemoglobin, the deformability improves both when oxygenated and when deoxygenated. And the point of sickling shifts so that they can tolerate less and less oxygen before they begin to sickle, begin to polymerize. And so hydroxyurea is kind of the ideal drug in that it's improving all of those metrics. It's improving your flexibility of your red cell when you're oxygenated, when you're deoxygenated, and it shifts the point of sickling to the left so that you can tolerate less oxygen before sickling. Transfusion does a similar thing. Of course, now you are a mixed population of donor and recipient. And we've begun doing the necessary clinical validation of the oxygen scan of the Lorca, which is the company that makes the ectocytometer I'm describing, beginning with kind of the low-hanging fruit of a retrospective chart review, looking at number of VOCs. Now, both my lab at Texas Children's and a lab in Utrecht in the Netherlands and a lab in France. Guys, keep it down, okay? Close the door. That's the downside to working from home. We all had been measuring our patient samples longitudinally on the Lorca. And so we could go back and look at our data on 2 years of patient samples, hundreds of patient samples run on the Lorca, and look and see whether there was a difference in these oxygen scan biomarkers between patients who had had a VOC in the last 2 years and those who had not. VOC being vaso-occlusive crisis or pain crisis. So one of the downsides to this collaboration was that my cohort was pediatric and their cohort was adult. I'm of course in the US, they're in Europe. And so there are likely some cultural and geographic and patient care issues. But what we did see was that in both of our cohorts, individuals who had not had a pain crisis in the last 2 years had a lower point of sickling, meaning they could tolerate less oxygen before their red cells became more poorly deformable. The data for the elongation index minimum and maximum was less significant, and I did not find a difference in the elongation index maximum in my cohort, although I did with the elongation index minimum, and they saw with both biomarkers. And so again, lower point of sickling or higher elongation index, either maximum or minimum, correlated with fewer VOCs, no VOCs in the last 2 years with this cutoff. So that's the beginning of clinical validation. It'll be important to look at other clinical complications and also to look at the data prospectively, longitudinally with the same subjects. So one of the uses of this device, again, is to analyze the impact of these novel therapies. And voxelotor, now called Oxbryta, has been FDA approved. We had looked at it both in vitro and in vivo. You can just add GBT440 to peripheral blood and get this effect. What you see when you either have a patient on the drug or spiking it in vitro is that GBT does what it says it's going to do. It keeps the hemoglobin in the oxygenated state, which shifts the point of sickling to the left, but doesn't have a major effect on the elongation index maximum. And again, kind of why would it? It may have a small, modest effect. Some groups see more of a change than mine did. Mine was pretty flat. But it does improve the deformability when deoxygenated, again, consistent with the mechanism of a drug that binds to the sickle hemoglobin, keeps it oxygenated, reducing sickling. So I had a concern when I heard about voxelotor and its mechanism that unlike hydroxyurea, for example, which increases hemoglobin, as does voxelotor, by 1 gram per deciliter on average, this is a temporary change to the hemoglobin. You've got a drug bound to the hemoglobin S. And if someone stops taking the drug will be cleared in about 72 hours in humans. And so you're going to have higher hemoglobin that is now unmodified. And the thing about sickle hemoglobin, which I kind of alluded to with that 21 hematocrit, 45 hematocrit comparison, is that when sickle hemoglobin rises, the viscosity increases exponentially. It rises much faster than a similar increase in normal hemoglobin or functional hemoglobin. So my concern was that we get this rebound effect of a rise in whole blood viscosity because we now had more of an abnormal unmodified hemoglobin quite suddenly. And I expressed that concern to GBT and proposed to give voxelotor to a sickle mouse model, the Townes mouse. and stop the drug and measure their viscosity. And they supported that research. And what we found, again, I have voxelotor in quotes because there's a variant of voxelotor that's metabolized much better by the mouse, and that's what I used. But just like voxelotor, the hemoglobin rose. So this is the same cohort of mice with control feed, with feed that has this voxelotor-like compound, and then the hemoglobin once we've stopped therapy. And so we got that same expected rise in hemoglobin, which dissipated when the drug cleared, but not entirely to control or baseline levels. And we measured the viscosity of 2 shear rates, kind of modeling venous and arterial shear rates. And we didn't really see a significant rise in viscosity, even though the hemoglobin was so high. And that was likely because, again, we've modified the hemoglobin, we've improved its quality. And so you can have more of it and not pay a viscosity price. And we saw a similar pattern at the higher shear rate as well. And here we did see a modest rise in viscosity when we stopped the drug suddenly, but far less than I had expected when I had been conceiving of this problem. Now, another way to think about viscosity is to think about the hematocrit to viscosity ratio. And this is important because it allows you to adjust for a higher hemoglobin. So you're looking at this ratio, which also correlates well with oxygen carrying capacity. And it's something that we are always thinking about when we think about not transfusing a patient above a hemoglobin of 10, for example. So even though the transfused blood is of better quality, you still want to make sure you're on the right side of that viscosity curve. You know, you can picture in your mind, and I'll probably include it in the slide at a later date, those U-shaped curves that you think about when you're dealing with polycythemia vera, for example. Hemoglobin rises, 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 oxygen carrying capacity increases as well. And then you get to this transition point where if your blood gets any thicker, it's going to start moving really slowly and your oxygen carrying capacity is going to decline. And this is something that the HBR gives you a good insight into. So whole viscosity is important, but it's also important to think about it in relation to how high a hemoglobin you have. And so for HBR, the higher the value, the better your oxygenation. And so here we see that the HBR is much better in the treated mouse. Again, they're well able to tolerate that higher hemoglobin because it's so functional. And then we see it begin to decline when we stop treatment. And the same pattern is seen with the HBR at the faster rate. So we're still seeing that our blood is actually more functional and better than it was before treatment, even after stopping the drug suddenly. So then what is the deformability of the red cell in these different stages? Well, of course, in the untreated mouse, the elongation index minimum maximum is reduced compared to the treated mouse. It's especially dramatic with the elongation index minimum. You can really see the drug effect, making the red cell far more flexible when it's deoxygenated. But what I found interesting was that I could still see some improvement in flexibility of the red cell even after I'd stopped the drug. And the drug had clearly already cleared, because you can see the point of sickling is the same in the 48-hour post-treatment as in the control feed. So the drug had dissipated. It was no longer shifting the point of sickling like it had when the animals were on treatment. But there was still some improvement in flexibility, and I found that very interesting. And I think it's something that may point to an added benefit of voxelotor in the red cell, perhaps antioxidant. I initially had thought it might be a hydration improvement, in part because GPT had kind of signaled that, but I didn't see any change in MCV or MCHC. I can't measure density with the with the ADVIA because of the size of the mouse red cell, but it would be possible to look at that with a phalate gradient. So even though the drug had cleared, the red cell was still not as rigid when deoxygenated as a non-treated animal's red cells. So this may also explain why we didn't see a worsening of the viscosity, because there was still some rheologic improvement lingering after the drug had been cleared. And we're looking at this at 2 additional time points to better understand the data, looking at 24 hours to try to capture more of that rise of hemoglobin, and at 72 hours to see how prolonged this deformability benefit is. So to conclude, this voxelotor-like compound behaved as described in patients taking voxelotor. Hemoglobin rose, viscosity did not significantly rise, and HBr rose. And off drug, while the hemoglobin dropped rapidly, we still captured at 48 hours a period where the hemoglobin was still elevated. The point of sickling had returned to normal, indicating the drug had dissociated. But the viscosity rise was very minimal, and the deformability appeared to still be better than at baseline. So another use for these tests is to try to develop a biomarker or something laboratory, wise to follow for these drugs that don't alter any conventional clinical laboratory tests like Endari. They performed their phase 3 trial using number of pain events in the last year as their primary endpoint, or hospital stays. And so that's a very unwieldy endpoint, and they need to do subsequent trials in order to satisfy things that the FDA had asked them to do and to try to get approval from the EHA in Europe, et cetera. So they asked me to help them find a biomarker that would be something they could follow in clinical trials. And unfortunately, even though I told them I think you should look at adhesion, they would only let me look at it on the Lurka. They were sure that was the answer. It's got to be deformability. Well, I ran samples both from patients before they started Endari and then on Endari, and also those spiked in vitro samples, trying to see what, if anything, Endari changed in red cell deformability in sickle cell. Compliance was terrible in my patient population. That certainly made the in vivo work difficult, but patients who reported good adherence and actually were filling their prescriptions and not turning away the boxes when they showed up at their house, did show an improvement in their elongation index minimum on the drug, and a slight shift of their point of sickling. Not to the same extent as voxelotor, and I feel that this is unlikely to be its major effect, but it is a statistically significant change produced by Endari. So to kind of take apart these different biomarkers, percent dense red blood cells are something that's been discussed for a very long time in sickle cell. Before the ADVIA, which of course has been around for quite some time, it was measured with phthalate gradients, and there were efforts to do the sort of clinical correlations that we're beginning to do with the Lurka. High percent dense red blood cell levels have been associated with higher incidence of skin ulcers, priapism, renal dysfunction. Certainly those dense cells tend to hemolyze more. If you have alpha thalassemia co-inheritance, you have less total hemoglobin within the cells, so your percent dense cells are lower, which may in part explain the different phenotype of alpha thalassemia patients. And dense red blood cells were used as a biomarker in a clinical trial in the late 90s for Seneca-Pac, which did reduce the percent dense red blood cells. But I always think about this trial when I'm designing experiments, because even if I think only one aspect of rheology is important or relevant, it's really important to measure all of them. Just like at first I was only going to do viscosity for the voxelotor trial, but then I thought, well, let's run it on the Lurka anyway, and that actually provided some important insights that helped me interpret the viscosity data. It's important to look at as many aspects of rheology and red cell physiology as you can, because they only looked at percent dense red blood cells for Seneca-Pac. And so in a phase 3 trial, they didn't see an improvement in pain crises. And it was very likely, I think most people would agree, kind of doing the postmortem of that trial, is that your dense red blood cells declined, but you had less hemolysis, your total viscosity probably rose, and the red cell quality was still pretty poor, so your HBr was probably still low and not terribly good. And so again, just looking at that one aspect, it wasn't enough to provide a clinical improvement. And so had they looked at a variety of aspects of red cell rheology, they may have better understood the problem and maybe have determined what role this drug might play in sickle cell care. For example, they tested in SS patients, whereas SC patients have more of a dense cell red cell dehydration issue. And with additional data, they may have said, oh, maybe it's better in the subtype. But since they only looked at SS and only dense red blood cells, they got a negative result, and it all stopped there. So you can do this phthalate gradient, which is quite time consuming and a little bit difficult to interpret, or you can run it on your ADVIA. And the two actually agree quite well. Mohan Narla did this work in '84. And so I prefer to stick with the ADVIA. And it's possible that other hematology analyzers measure this parameter as well. That's something that we'll be trying to figure out. Does the SystemX, which is used so widely clinically, also calculate this? Because it was interesting. I only knew that the ADVIA did this because Mohan told me it did. When I contacted Siemens and said, so where do I find this value? They had no idea about it. And I just had to kind of dig around through reams of data to find it. So it's possible that others do as well, and that's just not information that's made available because the focus has been, laboratory clinical use rather than research use for these devices. So adhesion, another very important parameter in red cell physiology and sickle cell. There isn't this, frequently used with, controls that are shipped to you like the ADVIA device to measure adhesion. This is something that's definitely a work in progress. There are a lot of different devices, a lot of different labs developing them. I initially was working with my colleague at U of H, Sergei Shevkaplias, and his device was kind of a compound adhesion deformability device. So his little chip had very, very small artificial vessels. They got down to almost single red cell microcapillary size. And initially they were all naked, they were unlined. And so you were capturing both the deformability because the red cell is squeezed through these tiny, tiny chambers and some element of adhesion, especially when you be unlining them with fibronectin and laminin. And this device actually was able to effectively distinguish between hemoglobin AA blood and hemoglobin SS blood, which might sound like it's something very easy to do, but it has been challenging for a lot of microfluidic devices. And so you can see that an SS patient, when you run their blood on a non-adhesive device, it runs more slowly. And when you then run it on an adhesive device, it runs even slower still. Whereas a hemoglobin AA individual, it doesn't matter whether it's lined or not. The red cells still just skate right through. So that's an important thing that it can do. But we're not uncoupling deformability from adhesion. So we're not really looking at the one in isolation of the other, which we may want to do for certain therapies and for certain usage. And it's also extremely time consuming, and any little clogging of white cells messes it all up. So, Sergey had to just say to me, I just don't think this can be used commercially and on a large scale. And I appreciated his frankness on that. And so I went looking for other things. There are devices that Umit Gurkin at Case Western makes that are lined with endothelial cells, which is excellent. But again, if you're trying to have a rheology lab in multiple sites, not every lab can culture endothelial cells, and it's quite technically difficult and expensive. So, having this great opportunity to work with Wilbur's group, he has a device that can be lined with protein or with cells. And my lab is back in Houston still is using it now. And getting great help and tech support from Wilbur's group. Be great to be in the same city soon. My lab is kind of coming in stages. I'm here, next person comes in about 2 weeks, and subsequent folks, three of my lab members are joining me here. And so this device, again, it needs to have the validation for the purposes I'm proposing to use it for. But it has been used to study sickle physiology and to look at the impact of different sodium concentrations, which is so relevant for our hydration efforts. So you may be asking, are these measures of red cell rheology good biomarkers? And what makes a good biomarker? Well, there's analytic validity. How well does this device perform? How reproducible are its results? If you run the same sample 3 times, 5 times, how close are those values? And that you can look at the coefficient of variability for that. It's excellent for the LORCA. The elongation index has a coefficient variability of 0.4. The point of sickling is a little broader at 2.2, but still very good. The dense red blood cells, excellent, 0.3. The adhesive device, again, this was done on the earlier one from Houston. I would, of course, want to work with Wilbur's device to calculate it for this. So what level of clinical validity do these biomarkers have? How connected are they to actual patient complications? Do they have clinical relevance? Well, the elongation index, minimum and maximum, again, it's a work in progress. I'm an author along with Minka from Utrecht on this paper, but we did see that low deformability values are associated with higher rates of sickle cell complications, particularly VOC. We've extended it out to some of the vascular complications like pulmonary hypertension. And point of sickling, again, a higher point of sickling at a higher oxygen concentration associated with higher rates of sickle cell complications. But further work should be done on this. Dense red blood cells is a bit of a mixed bag because of differences in methodology. But in general, most groups report that high values, high percent dense red blood cells are associated with higher rates of sickle cell complications, as well as adhesion being associated with higher rates of sickle cell complications. Now we have an anti-adhesive drug, crizanlizumab, which was shown in a phase 3 trial to reduce pain events. So that's quite good confirmation that adhesion, is a measurable biomarker that addressing reduces complications. So now that we have all of these drugs, a big question, one that I get asked a lot by these venture capitalists who are trying to decide what to invest in with GuidePoint. How are you going to decide which drug to use next? How do you prioritize these drugs? And that's something we're talking about as clinicians as well. What kind of algorithm do we have in our minds? And I don't really think that that's necessarily the right approach. Like, first I'll try this, then I'll try this. I think better, and probably what a lot of you here are doing, is looking at the patient and thinking, what is their clinical complication that's most pressing, most addressed, and choosing the appropriate therapy that way. Another way is to use these rheology measurements, these physiology measurements, for precision medicine. So run that patient's blood on all of those devices that I showed, the Lorca, the Hemox, the Advia, microfluidics, the viscometer, and see what is most abnormal, what is most out of range in that patient, and then pair the therapy accordingly. Of course, everybody's on hydroxyurea, that's understood, right? But what do we add on top of that? So if it's their elongation index minimum that is extremely low, voxelotor would likely address that. Point of sickling is also shifted by voxelotor. Dense red blood cells, that's a little bit stickier of an issue. I had done work for Sensilio back in the day. Some folks may be familiar with that. They were suggesting using their proprietary omega-3 fatty acid as a therapy for sickle cell. And it actually did reduce the percent dense red blood cells. Now, do you need to use their version, or can you just buy it from Costco? Probably can just buy it from Costco. But it did actually significantly reduce the dense red blood cells. And I think a better run company that didn't go bankrupt, we might have gotten a phase 3 and some further data on that. Another possible option is red cell hydration for reducing the dense red blood cell concentration. Adhesion, of course, crizanlizumab. Viscosity, a really tried and true therapy would be phlebotomy. And back in Houston, we phlebotomized, especially some of our hemoglobin SC patients who had very high hemoglobins, a lot of pain complications or retinopathy, and we saw a reduction in their whole blood viscosity and an improvement in their clinical complications. You then have to ask yourself, am I going to allow them to become iron deficient or not? And there's some issues with, of course, iron deficiency in children, so typically not, but those were some of the ways to address high viscosity. And so use these biomarkers to select therapy and then to assess the therapy. I mean, these new therapies are 100,000 a year, and for some, there's no laboratory change. Do you really want to keep them on this for 2 years and count their number of pain crises? Or is there a way to see whether we've addressed that rheologic abnormality in the lab? And of course, finally, I'd like to use these tests to assess gene-based therapies. The Cure Sickle Cell Initiative that NHLBI has had a grant opportunity where they specifically mentioned viscosity as or rheology, excuse me, as a possible endpoint for these gene-based therapy trials, and they invited applications for ways to determine whether a gene-based therapy had been curative. I submitted an application and it was funded. So me and two other labs meet bi-weekly to talk about our different biomarkers and the progress we're making in doing the analytic validation and the clinical validation. And so this is so needed for gene-based therapy because they're all different. We've got our lentivirus approach where we're inserting, lentiglobin genes throughout the genome and getting that type of hemoglobin. We have the CRISPR-Cas9 gene disruption approach where we knock out the erythroid element of BCL11A and get fetal hemoglobin production. And we'll hopefully in about a year have the base editing strategy where you use CRISPR to locate the sickle mutation, but then chemically change the base without cutting to create this functional hemoglobin macassar at the site of the sickle mutation. You just can't chemically change the mutation back to wild type. So this is a functional hemoglobin that seems to work just as well as hemoglobin A. So you have all these different strategies, and what do you have to assess them with? You can look at number of pain crises, which is a very blunt tool. Someone can have no pain crises for 2 years. I wouldn't call them cured. You can look at the percent hemoglobin S versus the percent functional hemoglobin, whether it's F or lentiglobin or macassar, but we don't really know what percent we need. There have been efforts to determine that for allo transplants, for our traditional transplants from a matched sibling or a matched unrelated donor. But I find this paper to be a bit suspect because here they're only using 3 patients and then they're doing mathematical modeling. And so, and from that they say, okay, we just need 20% chimerism. But that's dependent on a lot of assumptions. That these donors are all the same, and we know that the recipients are all the same. We know that the underlying recipients are extremely different. The donors are different too. Some may be AS, some may be AA. But again, these labs need to be interpreted with a grain of salt, and we need to functionally assess what that actual red cell product is. For example, a lot of this depends on how long that red cell survives. And if the recipient's red cell has a very short survival, you can have less chimerism because the donor is going to outcompete the recipient. But this is going to be different for different subjects. And now with gene therapy, we're introducing an even more complexity because we may have one allele edited and not the other. With lentiglobin, it's going to be a variable dose effect throughout the genome. And so it'll be possible to have a red cell that's still not very good quality, but has just enough of a functional hemoglobin to survive quite a while. And so that's going to throw off all of these calculations. So the unmet need is how to determine if a cure has been achieved, how to compare these multiple strategies head to head with something meaningful. And the biomarkers that I proposed in the application, I'd actually been collecting data from the Lorca, from the Advia, even from some of the microfluidic devices for some time. And so what I showed them was that I could give you a range of values for each of these biomarkers by genotype, and that these ranges didn't overlap. And what was really important about this hemoglobin SS group was I did not select them at all. They are of all ages, all therapies, all fetal hemoglobin levels, some on red cell exchange so that their sickle hemoglobin was quite suppressed, some with excellent fetal hemoglobin response to hydroxyurea, some very young with still elevated fetal hemoglobin, but they did not overlap at all in these biomarkers with a trait individual. So you can tell if an individual has SS regardless of any of the conventional therapies we have, or if they have AS. And what I proposed was that we set the cure to be a red cell population that is functionally in the AS range, in the trait range, because we know that's a generally asymptomatic carrier state. And this is asking more of the gene therapy than what had been proposed prior, which was just no pain crises in the next year or 2 years, which is just unacceptable. So the tests looked like they would perform and be able to distinguish that. More work, of course, needing to be done with the microfluidics particularly. So I had all this viscosity data as well, and I dropped that as a test because it did not achieve that separation by genotype. There was overlap in the ranges I got for trait and disease. And I think it's more related to the technology. So not saying viscosity has no role. It was very helpful for evaluating the GBT therapy of Oxalitor, but I don't propose it as a biomarker for assessing cure because I cannot reliably distinguish between a trait individual's viscosity and an SS individual's viscosity. So part of the steps, the milestones of that Cure Sickle Cell Initiative grant was to do analytic validation and then clinical validation. And I've done the analytic validation for all but the microfluidic device since I adopted Wilbur's somewhat later in the cycle, and it was less characterized than the other devices, and also to do clinical validation. And so I'm quite far along with the clinical validation, again, because so much data had been collected on the Advia, Lorca, and well, the viscometer, but I dropped that. So we can already look at some of the existing data and see how these biomarkers distinguish between the genotypes. Of course, it was whole blood samples run on the Lorca with oxygen scan and the Advia. And I worked with our statisticians at Baylor to do a receiver operating characteristics or ROC analysis, which sees how diagnostic the test is. It plots the true positive rate against the false positive rate at different thresholds that are determined by the statistician. And so it would say, at this value, your likelihood of having hemoglobin SS is this, true positive, false positive. And we included S beta 0 as well. So we had a lot of SS beta 0 subjects, 240. We had much fewer trait. I'm looking forward to finding a good way to collect more trait samples and more age and race matched hemoglobin AA samples as well to increase these numbers. But it was certainly sufficient to do the analysis. You can see that a significant percentage were on transfusion, almost 17%, and quite a few were on hydroxyurea. There were some overlap and some on both therapies. Of course, none of the trait or AA were on those therapies. So we looked at the biomarkers by genotype. And again, they are the elongation index minimum, maximum, point of sickling. You can calculate the area of the curve and your change from elongation index maximum versus minimum and other different parameters that are reported by the Lorca. My statistician used my viscosity data anyway, even though I said I don't think this is the answer. She plugged it through along with the HBR. We looked at the dense cells, and of course, not enough data on the microfluidics to include in this analysis. And so she determined her cut point. And you can see these tests had excellent sensitivity and specificity. I hope our next COVID tests can be as good because 94%, 97% for the elongation index minimum, in the 80s for maximum, the point of sickling in the 90s. And interestingly, the elongation index maximum minus minimum, which again, I didn't think was anything, has an excellent sensitivity and specificity. And these are just each test individually. You can imagine in combination how powerful these would be in determining whether a blood sample has trait functionality versus disease functionality. And these are the ROC curves that she generated. What you want is for things to be as close to this corner as possible. And you can see that the elongation index maximum minus minimum is the one that's actually the closest to this upper left corner, with the others still performing quite well. Viscosity, again, I guess it wasn't bad. It was in the 70s to low 80s. And the dense red blood cells performing, again, we use this one with MCV range of 60 to 120, which is reported. And that has a sensitivity of 82%, specificity of 92%. And again, these are the ROC analyses, and you can see again, dense red blood cells performing the best with viscosity and HBR. Still good, but not as good. And these are box plots, again, demonstrating how well these biomarkers distinguish between disease and trait. And again, these are all comers of disease, not untreated. For point of sickling as well, no overlap. Viscosity, again, you can see that there is some overlap between disease and trait, both for the venous rate and the arterial rate, as well as HVR, oxygen carrying capacity. Dense red blood cells, again, giving good separation between the two genotypes. So as proof of principle, I thought it'd be interesting to run some allo-transplanted patients on these devices and look at their red cell function. I had expected them to be pretty good. And I did get a surprise on one patient, and I do want to look at more in order to understand whether she was an outlier or whether this is happening more than we think. There was a 27-year-old who had received a matched sibling donor transplant when she was 16. This is her donor. He's AA, he's totally normal. I ran his blood too just to make sure he hadn't been mistyped or something. And this is her. She's already undergone transplant. She had 40% chimerism, and yet she looks just like a sickle cell patient, maybe one on hydroxyurea. Really not what we would expect from a transplanted patient. We would have expected something more like patient 6 here, or even patient 5, straight line or just a little loop. And when we talked to her more, she was having pain crises, was somewhat in denial about them and managing them with ibuprofen mostly. And her retic count was quite high and she was anemic. So she's this patient here. And unfortunately, the chimerism has been measured in all different ways. These two patients, despite, again, having a hemoglobin profile that looked quite good and acceptable for their trait donor, their rheology values fell kind of in this no man's land between trait and disease. And so they're much less far out from transplant and maybe someone to watch. So these rheology tests could be used to signal that there may be an upcoming problem. It may be more sensitive than looking at chimerism, certainly probably cheaper. And it may challenge some of our assumptions about how much reduced conditioning we can get away with, how much chimerism is appropriate, and look at this more on an individual basis rather than giving these blanket statements of this is all you need, which everyone happily puts in their grants and uses as a reason for why their strategy is going to work. So the next steps are to perform that same level of clinical and analytic validation of the adherent microfluidics that we'll be using. My lab is getting expert at it now, and actually I had been in talks with Novartis from the beginning about these biomarkers because I had asked them for a letter of support for the grant application, which they gave me. And then they kept asking me, are you funded? Are you funded? Because they wanted to put these tests into their clinical trial, their CRISPR-Cas9 based clinical trial of fetal hemoglobin induction. And they wanted to know if they had to pay for them if I got the grant, which is really funny coming from the richest pharma company in the world. But maybe that's how they got that way. So because I was funded by NIH, I didn't charge them for any of these tests. But of course, they don't own the data now either. So they're sending me samples from their clinical trial. It appears they've enrolled somebody because I've received the first sample, and I'll get another one tomorrow. And as soon as the MTA is finalized, they'll start coming to Emory instead of Baylor and Houston. And I'm really glad that Novartis wants their red cell product to be looked at rigorously, looked at this way. And Beam Therapeutics, which does base editing, wants to be analyzed this way as well. Bluebird Bio said no, Vertex said no. However, once those patients are edited, they're being seen at Emory, we can obtain a sample on a research basis and look at what the end product is. How sickle does that, does the blood circulating in their bodies look? So I think this is the future for how to compare head-to-head these different gene therapy trials. And I hope that all trials will aim for at least a trait level of function and correction. Another use, again, is to uh, assist in clinical decision-making, which of these novel therapies to give our patients. And along with Satish and David, I'd like to develop a red cell physiology core here at Emory, have these tests be CLIA certified, be part of the medical record with an interpretation from a board certified hematologist, as well as continue to use them with preclinical industry and academic projects as we have been. All right, so my wonderful lab, Celeste, Ankush, and Yankai will be coming soon. I'm so grateful that they have agreed to come to Atlanta. I think they'll love it here too. And I'm really excited to continue to work with Wilbur Lamb and of course my collaborators in Utrecht, and of course still with my colleague in Houston. And very grateful for all the funding and support. Excited to start on the 14th. All right, happy to take questions.
Hi, Vivian. This is awesome. Of course, I paid most attention to the, to your experiments in the transplant arena. So the gene therapy data so far suggests that the sickling and non-sickle hemoglobin are about 55-45.
Yeah.
And on the very limited data that they share publicly, it seems pancellular.
I don't believe that. We talked about this. Yeah, this is the problem. F cells are way too blunt a tool. It does not really, once your hemoglobin is quite elevated, whether it's sickle or hemoglobin A, it's going to look pancellular, but it really isn't.
He's allergic to it.
That's what he got the last time.
And I'd like to see whether Pablo Bertolucci's method of measuring hemoglobin F content is better. I haven't gotten to try it out yet, but he published in the American Journal of Hematology about a month ago. Some discussions have been that method, still needs some improvement, and but that's, I think, a more rigorous way, and that'll help us better determine whether something's truly pancellular or not.
Sure. Thank you.
I wish it was pancellular. That would be fantastic, but there's just no way it is, because you're not getting enough alleles corrected with the CRISPR strategy, and you're not getting enough insertion with the uniform insertion with the lentivirus strategy.
Vivian?
Yep.
I was thinking about the observation that even with the mouse red cells, after discontinuing voxelotor, and even at a point where the voxelotor should be long gone, right, from the red cells, that you still saw that the red cells were had better deformability in with the in the Lorca than what they were before. And you said something about you wondered if it was hydration, but you now think not. So then I would say, well, it seems like it would be most likely some residual membrane effect. I mean,
I agree. Yeah.
There's only three.
Yeah, there's a limited number of things.
Deformability is determined by three things, right? Hydration and intercellular cytoplasmic viscosity, and membrane flexibility, and then surface area to volume ratio. So do you have a hypothesis about why there might be a beneficial effect of voxelotor on membrane flexibility that would last beyond past where the drug is no longer present?
Yeah, so one thought, is of course that red cell has been having less sickling through its passage because it's had the drug bound to its sickle hemoglobin. And there are a couple of researchers at U of H that measure intracellular free heme. And of course, we think a lot about extracellular free heme, but not a lot about intracellular free heme. And I'm wondering if, due to the drug presence, we have lower intracellular free heme. That is, of course, again, adding to the oxidative stress and to the membrane abnormalities that we see in sickle cell. And I know their method. We could test this hypothesis and see whether there's a difference in the intracellular free heme in the presence of voxelotor and whether it persists after the drug has been cleared. We can use patient samples for that. We can certainly spike the blood for the drug present, but of course you would need to get a patient sample for the drug cleared type of sample. But you could at least quickly test the hypothesis with an intracellular assay of just adding voxelotor to a patient sample and see whether that alters the free heme the intracellular free heme concentration. You'd probably have to incubate it for some period of time.
Vivian, another possibility is improved surface to volume ratio. Is one of the things that happens with sickling is the spicule formation and loss of membrane fragments from cells. So that would be sort of top on my list of one of the things, as well as the oxidation question.
Yeah, I think oxidation is a very good possibility. We talked a little bit about the difficulty of measuring that. Now, the surface to volume ratio, I guess to me that's related to hydration, and I would hope that I would see some signal there.
Well, not necessarily. I mean, if you lose surface area, you're going to lose surface to volume ratio, even at the same volume. So that's a potential possibility. It's hard to measure. We can talk about how to do that.
Yeah, and of course I don't have dense red blood cell measurements on the mouse blood. That would have to be done with.
Well, they tend not to have very many dense cells. The mice don't.
Interesting.
They don't. The cells don't seem to live long enough to get dehydrated. So
yeah, there are definitely some limitations to the mouse model. Maybe some of our patients will kindly produce some off-drug samples for us.
Yeah, well, no, I think you're likely to see that at some point. Yeah. One other follow-up question is whether or not you have actually tried to look at in vivo reducing or in vitro reducing agents on the behavior of the cells in the Lorca.
Yeah.
Because, we can reverse some of the transport abnormalities with as simple a treatment as dithiotrietol for a small incubation. So it'd be interesting to try that in vitro and see whether or not you improve the elongation index at baseline.
Yeah. I think writing up a proposal of these various suggestions and offering to do that at GBT's funding would be great.
So Vivian, just not to beat this up, but the osmotic gradient ectocytometer, that technique, osmotic gradient ectocytometry, was originally developed in part to tease apart those three independent mechanisms that impact red cell deformability, and certainly with the surface area to volume hypothesis that Clint has, is there a reason you couldn't just use the old osmotic gradient ectocytometry to sort of try to look at that, or does that not work with sickle cells?
Well, yeah, you can certainly you can uncouple the Osmoscan from the Lorca and just run it as a standard ectocytometer. You can certainly do that. The issue is that I really only saw a significant difference with elongation index minimum, which is the deoxygenated red cell. I don't think I would detect anything running the Lorca as a standard, just regular Osmoscan, or Osmoscan, not oxygen scan. Because that wasn't where the remaining benefit lay. It was in when we deoxygenated the red cell.
Well, Vivian, although this is your second time giving you this talk in a week, this was once again a great talk and clearly a very engaged audience. I thank you all for your questions and attention. At this point, we will have to wrap our session, but of course, we will see you back in a couple weeks for our next ARC session. And again, join me in welcoming and thanking Vivian for her talk today. Bye, everyone.
Thank you all so much. I can't believe you listened to it a second time. Much appreciated.
Bye, everyone.